Okay.
Maybe you said it, but I might have missed it. How distant in time were the samples? How?
Sorry?
The samples, some of the sequences that you looked at. I didn't understand the date of collection. So were they collected, very distant in time, or?
No. Basically, what we have at first is the different location of samplings and the exact date of sampling. For the location, we need the coordinates, exact coordinates of the virus to be able to detect the effect of environmental and human factors on the transmission, and the time just to make us able to reconstruct a time-scaled tree for the transmission of the virus.
Yeah. But I was just wondering if the sequences were obtained from viruses collected at very different periods of time. That might explain why you have such a disconnect.
We've got some from 2000, 2013, and some way older. You can see there. We've got some from quite recent, from 2010, and other way older, from 65 or from the 80s.
Tatiana, I have a question for you. It's regarding those prevalences in the cattle and buffalo, and you make a statement that the SAT2 you found to be least prevalence in Tanzania area, but we also know that SAT2 caused most of the outbreak in Southern Africa, where I'm from, or South Africa, Botswana, Zimbabwe. Do you think this is something that is it virus related? Will it differ from region to region? Maybe what factors are involved in this prevalence?
I mean, SAT2, from what we know, it circulates in both cattle and buffalo. In Tanzania, that is we have very limited results on the buffalo, unfortunately, and it's all kind of based on serology. But we know for sure that there is a lot of SAT circulation in cattle, and it seems to be independent from what goes on in the buffalo. So, we can't say much about the directionality of transmission, but kind of the level of infection in cattle, independent of buffalo, is so high at this point in time that while there could be some transmission in some areas where there is a lot of close proximity, it doesn't seem to be the main driver. And also, I don't know, the sample sizes are also very different. Although the VNT results that I showed were based on consistent sample sizes between buffalo and cattle.
Any more questions? Nick?
On buffalo cattle transmission, etc. I think as everyone sort of realizes that buffalo or carrier to susceptibles is a very rare event, and I don't think we can hope to replicate that experimentally, because in nature they have hundreds, thousands of animals, and this only happens occasionally. So when you try and do it with 10 cows and 10 buffalo in an isolation unit, you're not going to, people have to wait till, for years for it to happen. But clearly in nature it happens. So I think that the best way of looking at it is to look at the data for natural transmission. And there is plenty of data to show natural transmission. And one of the best bits is SAT3. SAT3, there is no known cattle outbreaks of SAT3 anywhere. When there are outbreaks, they have to come from buffalo. Good. Sorry? Yeah, well, that came from buffalo, though. Yeah, I think we do. I would say yes, we do. But anyway, you can show that there are many introductions of SAT3 from buffalo into cattle, and I think there's good evidence that it happens. It's also, you can see it in Southern Africa where FMD has been well controlled, but you probably won't see it in places that, in like East Africa, where the virus is endemic, and it's probably not going to be an important factor for initiating new outbreaks generally, whereas in a place where it's well controlled, it will be important. So I think that's really what I wanted to say.
Thank you. Elizabeth?
I have another question for you, Nick. When you show those recombination data, I look at the O's, A's, and C's. They seems like the capsids are very uniform, but when you look into the SAT, there are spots all over where you see fragments of between the different SATs being represented in all of them. So there is a little bit of something going on the capsid. Do you have any observation or any comments on that, or something that struck your attention?
No.
But if you look at the graph you show, it really has spots all over between SATs. I don't know if you still have it.
Is it possible to show that presentation of Nick? Thanks.
Sorry.
Okay.
Here, you see on the...
So it's one before after that one.
This one.
Yeah. So what are those where you have in the capsid?
Yeah, I'm not sure what's going on here exactly, but I mean this.
There's also the one around the purple substance, which you don't see in the orange, blue, or gray.
Yeah, I mean.
These are within serotype, though, because on the previous slide it showed colour by serotype.
Yeah.
Oh, okay.
So by serotype you don't see.
Oh, okay. All right. I got confused. I thought it
Yeah.
was within serotype.
Okay.
Thank you. Anything else?
Yeah, so actually I have a question for Nick.
Okay.
So I feel a bit silly doing this when he's about 2 foot away. When you do go to the East African ones, I was just wondering whether you actually find East African SATs that are like the rest of Africa, or do you only find the Queen Elizabeth ones? Do you only find those ones in Queen Elizabeth, or do you find other ones as well?
No, you only find those in Queen Elizabeth.
Right.
They're completely separate.
But yeah, so and whatever the other area was, there's just exclusively that topotype within.
Was East Africa more generally. So, but the Queen Elizabeth is only the Queen Elizabeth National Park. And I mean, the fact that SAT1s are interesting in that they, as far as we know, didn't occur in Tanzania, Kenya, etc. up until 1971. They occurred in Uganda before that, but all the SAT1s in East Africa now, at least from Kenya downwards, came up from Southern Africa in 1970-71. So there will be some links.
But none of them made it into Queen Elizabeth National Park.
No.
That's amazing. Anything else? Can I ask, I know Jadendra had 2 more slides that he skipped. Jadendra, do you want to say something about that information on your slides? Okay.
Oh, okay.
Yeah, this antigenic relationship, sorry for that, I couldn't explain it properly. So what he found is that with respect to the March collections, because the outbreak happened in Jan, in March collections, all the viruses, apart from one, AC means asymptomatic and C for cattle, So all the viruses, almost all of them has a R value in the 2D MNT is more than 0.3. While in case of buffalo, during the course of the infections, we found that during the end period of the course of study, there is a significant antigenic variation. They are not antigenically divergent in nature during the November and February collections. So which is exclusively found in case of buffalo, but not in case of cattle, apart from one cattle. So what we believe is that antigenic diversity or antigenic divergence during the course of persistence is more in buffalo as compared to the carrier with respect to the data from a particular farm. And when we analyze the sequences from those animals which are being antigenic divergent as compared to the acute phase animals, we found there are many sites, 1, 2, 3, 4, 5. But the blue color one, that BP154, BP1143, and BP3131 are on the surface. They're on the exterior sides, while the rest of them is in the interior sites. So what we believe is that these sites may be important for the antigenic diversities. And currently we are in pursuit of the reverse genetics approach to find out whether these are the new putative epitope responsible for antigenicity. And we found, with respect to the BP1143, we found some of the preliminary data which shows that whenever there is a changes, the particular changes in BP1143, the animal, the particular virus is having antigenically divergent as compared to the normal viruses. So similarly, if you, how to go down and back? Sorry. Yeah. Similarly, one of the significant findings is that in case of March, one of the animal, AC means that asymptomatic cattle, during March collection, the R value is 0.23, while in case of May collection, it become 0.8. Which you found when we thought out what is going on there. Then when you did a structural modeling, we found that during the March collections, the changes in BP2194 is the alanine to proline. As you all know, that proline is a helix breaker. And similarly, and at BP3166, there is changes from the aspartate to glycines. And both of these sites are related in the internal site. So while in case of the May collections, those are changes to means P2A and D to G. So what we think is that although they are present inside, but they provide some of the structural flexibility during the May collections, as a result of which the epitope has been exposed on the surface and giving a R value more than that. While in case of the March collections, because there is a helix breaker is there, so we believe that due to that changes, the R value remaining less than 0.3. These are the speculation, and based upon these information, we are doing the synthetic virus approach or the reverse genetics approach to prove whether the hypothesis is correct or not.
Dawn, I think you first.
So Jitendra, forgive me if I've not clearly understood what you're saying. So are you saying that there's an antigenic shift in the viruses that are from the carriers?
Yep.
And you're saying, are you saying that essentially they are viruses that are escaping the antibody immunity that's already in those animals? So those animals have generated a good antibody response to the clinical phase of the disease, and basically what you're recovering in the carriers are viruses that essentially are much more able to escape the antibody that's existing in those animals.
Yeah, these MNT data we are comparing with respect to the acute phase virus and the virus collected during the persistency. So during the initial phase, in case of buffalo, as you can see the chart that the R value is more than 0.3, while in case later part of during the period of persistency, there is antigenic variations. And that is in case of buffalo. So we believe that during the phase of virus persistency, there are viral activity as a result of which we get antigenically divergent virus. So the main take-home message is that what we believe is that there is viral activities going on. It is not the virus remaining in the quiescent stage during the time of persistency, but there are some kind of viral activities going on during the time of persistency.
Okay, Louise, I think you have something to add, and then we'll go to Sajja.
So, Jitendra, just to clarify, your neutralization to calculate your R value is not based on the current serum from the animal at the time you recovered the persistent virus. You're doing this, right?
Yeah.
So you're not measuring what you're asking. That's not the case. He's talking about that the virus
with respect to the
itself
vaccine strain
has a lower R value. Okay, so it's not... Right. Anyway, that's not. Just wanted to clarify that.
Okay, Satya, you have something to ask?
Jitendra, I am having one question. So when you started the animal experiment, whether you checked all the animals, they are not carrier at all, because it is an endemic country. So have you checked this one? From probing sample, from everything?
No.
So then how, whether you are thinking this virus is from the outbreak, or it is sitting maybe few months or few years before?
Yeah.
Because you are getting two types of virus. That is one question.
Yeah.
Then the second question is, you got also in some animals it is more changes, some animals less changes, or you got whatever changes you got, it is persistent. It is the same for all buffalo carriers, or only for few carriers, or not for true for other carriers?
No, the changes with respect to nucleotides you are saying, or?
Nucleotide or just it for both carrier animals?
No, what I'm going to say is that I missed that one of the slide. There is no fixes on amino acids between the different stage of the sample collections. And with respect to your first questions, if you see that the parsimony analysis, all the viruses which has been isolated from the carrier animals, they are grouped together, apart from the 6 isolate, which have a nucleotide sequence divergence more than 1%. Yeah. Yeah, we have it.
Okay, thank you so much for this discussion. I think we're going to stop here. There's just a few announcements that I have to make. One of the announcements is that the dinner tonight has been moved till 7 o'clock. It's not 8 o'clock, it's 7 o'clock. And please, you have to bring your cards with, otherwise you're not going to allow it into the venue for the dinner. Is that correct? Okay. I have no idea.